Thank you, Matt. Our last speaker is Professor Alison Van Eenennaam. Alison is a Distinguished Professor of Cooperative Extension in Animal Genomics and Biotechnology here at UC Davis. Her research focuses on applying DNA-based technologies to livestock production systems and the development of genome editing approaches for ruminants. She's an award-winning communicator, having delivered over 800 global presentations on agricultural biotechnology. Welcome, Alison.
All right. Thanks very much, Rob, and thank you so much for the invitation to present here. And welcome to Davis. It's certainly put on the weather for the international guests. So I was asked to speak about how genome editing of the host genome could play into reducing methane emissions, or whether it can or not. And I kind of like this slide. It's a world-famous Sydney Angus bull from the Ball Blair Ranch in Australia. And it was the winner of the Sydney Royal in 1961. We've changed a little bit since then, and I think it's pretty interesting to look at the difference you can make in only 30 years when you have a phenotype that's very easily measurable, that is height, and also fairly heritable. And looking at Troy's pull and push between the packer and the cow-calf operator, maybe the 50s is more of a cow-calf operator efficiency and the 80s is more of a packer thing. But why they changed so much was because selection at that time was on height, and maybe we ended up getting a more moderate framed animal because you couldn't actually see the breeders anymore in the 1980s picture, and they didn't want that to happen, so they had to get it down below head level. But we have actually developed these balanced selection objectives now where maybe height isn't the only contributor to overall profitability, and maybe there's some other things that play in there. But you can make pretty big changes using conventional selection. So that is selection based on naturally occurring genetic variations that the breeder's eye uses to then select the parents of the next generation. So we've got a new tool that's come on the scene, and that's the genome editors, most famous of which is the CRISPR-Cas9 system, but there are some others like Talens and zinc fingers. At the end of the day, they all do exactly the same thing, which is make a targeted double-strand break in the DNA double helix based on where you tell it to cut. So there's 3 billion base pairs in the bovine genome. I can tell this pair of scissors to go cut at the myostatin gene on chromosome X or wherever I want it to cut. And what happens after that happens is it will be repaired by the cell's natural repair machinery. So DNA often breaks during recombination, so there's enzymes in the cell to repair that break. It can be it can be repaired in one of two ways. It can go down what's shown here as the left-hand pathway, which I call the knockout pathway, where you get a small deletion or insertion of a base pair or two that throws that 3 codon reading frame out of kilter and effectively inactivate a protein. Why would you want to do that? Or maybe the protein is something you don't want expressed because it makes your animal susceptible to disease, for example. Or you can provide a template, shown here in the blue, that is homologous to either side of that double-stranded break, and it will introgress this blue piece of DNA. That might be used to introduce an allele from one breed, for example, Senopol, the slick allele, into, for example, Angus to make slick temperature-controlled Angus that are better able to cope with high temperatures. So that it can be used for intra-species or intra-breed allele movement around between breeds maybe that don't have that allele. It technically could be used also to make transgenic or bring DNA in from another species, and that's kind of more traditional GMOs, I guess, for those of you that know that language. I think the thing that is attractive about particularly these edits is there's no foreign DNA involved, and as a result it doesn't generate GMO regulation in a lot of the world. So if we look at the products that have been produced for food animals globally that have gone through a regulatory evaluation in at least one country that could technically be commercialized, that's my way of saying they're not always on the market, but they are in the case of the fish in Japan, what you'll see is there's a single gene that's been targeted in each of these applications, either to turn them on or introduce an allele, and it's targeting things like growth traits or more recently and very pleasingly, the approval of the disease-resistant porcine respiratory and reproductive syndrome pig that was just granted approval by the FDA last month, which is part of Genus's pig improvement company, and a very exciting trait to be first out of the gate. My point is, there's a gene that's involved. So is there a gene that's associated with methane? Is there the methane gene? We can just go knock that puppy out and we'll be done. That's how it's going to work. It's a silver bullet, right, Rob? Yeah, not exactly. So when we actually do GWAS, which is genome-wide association studies, looking for genes that are highly associated with this particular trait, we get kind of a scatter plot. So these are 3 different methane emissions per cow, per kilogram of milk, so that's more emissions intensity, per kilogram of fat. We get kind of some maybe peaks occurring, so across the x-axis is all the chromosomes, and the higher the peaks, the more significant that some little area of the genome is. And you might get excited by that, but actually, if you look at more other traits, for example, height, which is highly heritable, what you'll see here is the red line in this is the same as the red line here. And so basically, this is almost background noise in this graph, and the ones that are really big that we know have huge effects, like PLAG1, jump out at 10 to the 100 here, whereas over here we're at 5. And so there's just no giant effect loci that have jumped out and said, here I am, knock me out, and I am going to be a methane-efficient cow. This is what's called a quantitative trait. Many genes are going to contribute to that phenotype, and that's best suited to using tools like genomic selection, which actually enables you to sample the entire genome that animal inherited and then make your prediction equation based on that, because it does take account of the thousands of genes that likely affect that. So I guess to me, genome editing is a useful tool. It's the cherry on top of the breeding sundae that's shown here, but actually we need our breed associations to develop our selection goals and objectives, performance recording, progeny testing, AI, ET, somatic cell and nuclear transfer, cloning, genomic selection. All of that makes up the ice cream sundae of animal breeding. And yes, we could bring in a useful trait here and there using genome editing, but in my opinion, it's not going to be a big trait that's going to go in there and do methane, because it's going to be a complicated trait. And I hope that I have kind of position genome editing as part of kind of a suite of tools that we're now using to accelerate the rate of genetic gain towards our structured breeding program with a clear breeding objective. That is, we are going to use all of the advanced reproductive technologies, genomic selection, and where appropriate, we might bring in qualitative traits like not growing horns or a coat color characteristic using genome editing. But as of now, it's less suited to dealing with these what's called quantitative traits, and that's really best left to the purview of genomic selection, which requires these large phenotyped, genotype reference populations. And that's really where the bottleneck is to make these genetic predictions that we can use to select animals for this trait. So thank you very much, and I look forward to the discussion.